My name is Jack Darcy, and I'm a PhD candidate here at CU-Boulder's eBio department. My work focuses on better understanding microbial communities through a big data perspective. My research takes place primarily in high-altitude sites next to glaciers, where we can understand microbial communities and how they develop through space and time simultaneously. My research takes me to pretty cool sites all over the world. Like I said, most of them are near glaciers and glacially related sites. One of my favorite ones that I'm going to talk a bit about today is a site in the Peruvian Andes at about 18,000 feet above sea level. We collect soil samples from each of our sites so that we can grind them up and then extract DNA out of them. We use that DNA to figure out what kind of different microbes live within the samples. All of our soil samples have to be stored in these big -70-degree freezers like this one, because the boys at the USDA are pretty serious about the storage of foreign soils. So it has to be kept under lock and key. And here we can see some of the soils we've collected. This big bag of dirt was collected from 6,000 meters above sea level, which is pretty high up. We use that soil for other projects. That one in particular is to figure out how life lives in the really extreme environment on top of those volcanoes. This is my research site in the Peruvian Andes. It's the Cuckoo Glacier, and it's at roughly 18,000 feet above sea level. It's been retreating pretty quickly over the past 15 or so years. We can see repeat photography of that site between 2005 and 2010. And if you look at the little person for reference in both panels, you can see that the glaciers move back quite a bit between those two time points. This picture I took in 2015, and we can see that the glaciers moved back even farther and has melted into a little pond in front of it. In the foreground, we can see the site where my field plots are. At the sites in Peru, I used an aerial drone to help me map out where our different plots are next to the glacier. This is an aerial view of those plots. And as you can see, there's 4 different treatment types. We've got control plots, plots where we added phosphorus, plots where we added nitrogen, and plots where we added both nitrogen and phosphorus. Nitrogen and phosphorus are both common contaminants from both agricultural and industrial processes. So part of understanding this site and its ecology is understanding how those compounds can potentially influence how the microbes at this site naturally develop into communities after the glacier has exposed them by retreating. The drone allows us to visualize where the sites are in relation to each other. And for scale, here's my PhD advisor taking a picture of the drone in the corner. Standing at the glacier and looking back towards the lake, we can see the glacial meltwater draining back into Laguna Subinacocha. The lake is basically formed by the meltwater from the Cuckoo Glacier. And the lake is really cool in and of itself. The glacier is draining till down through that valley into the lake. And that's providing nutrients for the lake. So the kind of greenish color comes from like algae and cyanobacteria that are growing in that lake. But the glacier used to cover this entire area. As it retreats, it's depositing things back down into the lake as a result of weathering. But any pollutants that wind up on the glacier, therefore also wind up in the lake. So understanding how communities develop right at the tip of the glacier as it retreats is important to understanding perhaps effects on the lake as well. At glacial sites like mine, we can see how microbial communities develop from a very interesting perspective, because since the glacier is moving over geographic space, we can look at communities at different time points as it's retreated. This gives us really valuable insight into how communities actually develop, so we can hopefully come up with better, more generalizable trends. In the future, my research is moving in the direction of genes instead of just different microbial species, so we can know exactly what traits are being selected for as the glacier retreats and as the communities develop.